Okay, in the third video, we're going to look at the last phase of photosynthesis, which is the synthesis part. It's also known as the Calvin cycle, and historically it was referred to as the dark reactions. And that was only called the dark reactions because the first part of photosynthesis gets called the light reactions. We're actually trying to phase out the use of this word dark reactions because it implies that this happens in the dark, which is not necessarily true. It doesn't require dark, it just doesn't require light, which might mean that dark reactions is an okay name, but I find it to be misleading, so I think it's better to use the word Calvin cycle or the synthesis reactions. So these were the two stages. In the first stage, we made these energy intermediates. Nobody cares about oxygen, well, except for all other life on the planet, but we made these energy intermediates, and now we're going to use them to synthesize glucose or other carbohydrates. Okay, so the Calvin cycle is the one that does this. This is going to happen at the stroma of the chloroplast. So let's look at the chloroplast again. There's that outer and inner membrane, and then it's got these, I'm going to draw it in green, it's got these thylakoid membranes, and we talked in the last video about how during the light reactions, you end up building up a lot of H plus inside here, that we concentrate H plus inside the thylakoid lumen, but then outside the thylakoid lumen, we're doing things like making ATP, because as that H plus comes out, we generate ATP as ATP synthase spins. And then we also, in those light reactions, we made an NADPH. So out here in this area, this is called the stroma of the chloroplast. This is where we're going to use these guys in the Calvin cycle. And so in the Calvin cycle, we're also going to take the carbon dioxide from the atmosphere, and we're going to actually put it, this is where we're going to fix the carbon. So we're going to take 6 carbons from the atmosphere and combine them with 12 water molecules, because that's what's required to make C6. We're going to need 6 carbons, H12, so we're going to need some hydrogens, and O6, we're going to need some oxygen. In this process of getting these guys fixed into the glucose molecule, so you saw if we kind of added up the number of oxygen we have, well, we have 12 oxygen here, and we have 12 oxygen here, but we're only going to be using 6 oxygen for glucose. We're going to have those leftover oxygen molecules, and so those are accounted for over here. And then same for hydrogen, so we're going to account for those as well, and so that's why we're seeing these water molecules moving here as well. And we could simplify this equation, but this is more of the full equation kind of written out. And then also in this process, we're going to be needing to use a lot of the ATP and NADPH produced. So, that those light reactions have to go on quite a bit of times in order to get enough of that ATP. But we're going to be using that ATP in order, because this is an endergonic reaction, so we need to have some energy input. And so these energy intermediates will be our endergonic input in order to synthesize the glucose. So this is going to happen in 3 phases. We're going to look at each of them sequentially. The carbon fixing phase, then things are going to be reduced, gain energy and be reduced. And then finally, we'll see those carbohydrates are produced. So the Calvin cycle is going to be a cycle, which means we're going to regenerate what we start with at the end of the cycle. And so you're not seeing how we're regenerating it yet. So you'll have to trust me for a minute that we're going to be regenerating this molecule, which is called ribulose. Ribulose is a 5-carbon molecule. So we're going to have to regenerate it by this reaction. So for right now, just take my word for that it exists. And so all of this is within the stroma of the chloroplast. So this molecule is just kind of hanging out in there. It's like, oh, hey, I'm ribulose biphosphate, just hanging out here. It's got 5 carbons. And it's going to, first, it's going to combine with a carbon dioxide molecule. And so when it does that, the 5 carbons get added to the 1 carbon of a carbon dioxide, and it makes a 6-carbon molecule. But this 6-carbon molecule, as soon as it is made, it's so unstable that it immediately splits into 2 3-carbon molecules. So that's the first thing that happens. Now, mind what we're seeing in this reaction. Everything we're taking times 6, And that's going to be relevant because ultimately we need 6 of these carbons to make the 6-carbon glucose. So that's why everything is multiplied by 6. But I kind of prefer to look at it as just following this 1 carbon. But that's why you're seeing it written up here, because that's probably more technically correct way to look at it. But so this 5-carbon molecule combines with 1 carbon from carbon dioxide to make a 6-carbon molecule that immediately splits into 2 3-carbon molecules. And so here are those 2 3-carbon molecules. So it's a 3-carbon molecule called 3-phosphoglycerate. And what's written here, it's 2 3-carbon molecules, but times 6, because all of this has to happen 6 times to get to the 6 carbons of glucose. So that's why it's written here as 12. Okay, so that's our first phase. This is called carbon fixing, because what we're doing is we're fixing this carbon from the carbon dioxide into this 6-carbon molecule that immediately splits and becomes the 2 3-carbon molecules of phosphoglycerol. The enzyme that does this job is called rubisco. Okay, in the next phase, it's going to be the reduction phase. So phase 1 was carbon fixing. We just fixed the carbon, so now I got stuck in there. And this next phase, we're going to reduce. So when you reduce, remember that means gain energy or electrons, which commonly look more like hydrogens, in order to be reduced. So in this next phase, we're going to see this input of energy. So we're going to input ATP, and we're going to input the energy we had from our NADPH molecule. It's actually going to give up its electrons and its hydrogen and just go back to be doing NADP+, which can then be recycled and reused in other reactions. But so this molecule, it is losing electrons and being oxidized so that the 1,3-biphosphoglycerate can gain electrons and be reduced, which is what's happening in this reduction reactions. So we're going to use our energy intermediates in order to generate the next part of the process, which is going to be glyceraldehyde 3-phosphate. And if that sounds familiar, yeah, it was part of the cellular respiration reactions as well. So we have now formed it. So we're building glucose, and that's pretty far along those steps in order to make glucose, to make the G3P, glyceraldehyde 3-phosphate. And now we have used those energy intermediates. They have given up their energy and gone back to being oxidized or no longer having that energy, so that it can be now in this molecule that we're forming. So we've transferred that energy in order to make glyceraldehyde 3-phosphate. The third phase is called the carbohydrate production phase. In this phase, we're going to regenerate what we started with. Remember, I said I promise we'll get back to this 5-carbon molecule. So we're going to regenerate that 5-carbon molecule we started with. And then these G3P molecules, some of them will go off to become glucose. And so this is another reason why we had to take this whole thing times 6, because some of these glyceraldehyde 3-phosphate, so we got to 12 because remember we doubled it and we multiplied it times 6. So some of these molecules, 2 out of 12, will go off to actually form the glucose. The rest of them that are the same thing, the G3Ps, are going to be used to regenerate that 5-carbon molecule. And in order to regenerate it, there is going to be another energy input. ATP is going to be put into it to get that phosphate back to kind of destabilize the molecule to make it ready to accept or to fix another carbon into the molecule. So that's our energy input phase that enables that carbon to then bind with ribulose biphosphate and cause it to split like we saw at the beginning of this cycle. So this is the Calvin cycle. Yeah, I skipped a lot of details on it, but I think it kind of gives you enough of the idea that you can move forward and kind of feel like you've got a rudimentary understanding of this process. Basically, we are going to use some of this G3P to actually make glucose. So this would be different reactions that we could follow to see how we use G3P. Basically, it's 3 carbons plus 3 carbons, and we're going to get to glucose. But you and I both know it's not going to be quite that simple. But we'll use those to make the glucose, and then we also have to regenerate it so that we can run the whole cycle again. So that's the Calvin cycle in a nutshell, or maybe that's the Calvin cycle in the chloroplast would be a better way to say it. Okay, so now you've seen photosynthesis. You've seen both the photoreactions and the light reactions. I want to jump back real quick to the photoreactions because you might have missed it, but there is actually a problem with how I describe this. So looking back at the photoreactions real quick, remember, we harvest the light energy, and then we excite an electron, and we pass that electron down this chain, pumping hydrogen as we do so. And we're going to, in this process, we're going to use the hydrogen to generate ATP via ATP synthase. And we're going to use the electron that we're passing to reduce NAD plus in order to make our reduced NADPH molecule. And both of these guys go on to the Calvin cycle. So this is what we've looked at. And now let me tell you what the problem is with this. The problem is in the Calvin cycle, we actually use more ATP than we use NADPH. I don't know if you caught that, but that is a fact that we're going to use more ATP. So this is a problem because we're relying on the photo part to make the right amounts of ATP and NADH so that we can run that Calvin cycle appropriately. So the question is, how can we get enough ATP in order to drive that Calvin cycle? Because the linear flow, what we've just looked at so far, the linear flow will make equal amount of NADPH and ATP. And we actually need more ATP. So how can we use this idea and actually generate a lot more ATP? And so the answer, it kind of also explains a little bit why we need PS1 as well. But the answer is, instead of just going linearly with the electron where we pass it down and there you go NADH, and we pass it down and there you go NADH, and we make equal amounts of NADH as ATP. Instead of doing that, we can do this cyclic plan, which is, instead of, after you get to PS1 and you have that electron, instead of giving it up to the reductase and forming this right away, what if we just pass it back to one of the earlier players, the one of the plastoquinons? We pass it back. And the benefit of passing it back that way, instead of passing it forward to its final electron acceptor, the molecule that will be reduced, the benefit of passing it back is when we do so, we can then pass it forward again and use it to pump more hydrogen. And when we do that, when we pass it back and use it again to pump more hydrogen, we get hydrogen here again, even more. And this hydrogen is useful because we can use it to make even more ATP. So this would allow us to make less of the NADPH and more of the ATP, because the Calvin cycle actually needs more of the ATP and it needs less of the NADPH. So it can do this cyclic transfer in order to up the amount of ATP, because it's going to make that ATP because every time it passes the electron, it pumps H+, and we end up concentrating H+, kind of behind this dam, behind this wall, so that the only way it can get across, which of course it would want to get across the dam, because this is having it built up here, that's a lot of potential energy stored up here. The only way we can get it through is to let it go through this little trickle water wheel, where when it goes through, it spins the wheel. I'm spinning the wheel in the wrong way. It spins the wheel and smashes ADP and phosphate together to make ATP. And so that's a little trick that is used in order to equal out, or to make more, really, ATP, which is necessary to drive that Calvin cycle. So the chloroplast actually changes which way it favors. So the chloroplast favors the linear transfer. If it's got a lot of NADP+, then it's like, oh, well, I need to be reducing this. But once again, it's not cognizant. It's not thinking, oh, I need to be reducing it. It's just, look, there's a lot available. So look, we're going to do a lot of this. But if we don't have a lot available, then you kind of get to a point where it's like, oh yeah, I'm passing the electron. Oh, you're not here to accept it? Oh, where should it go? I guess it goes back here. And we do this cyclic thing. So if there's no NAD plus to accept the electron, then we can pass it back. And in so doing, when it goes back, it can be used to pump more hydrogen, build up more hydrogen, pass the hydrogen through ATP synthase, and make more ATP. Isn't that cool? So this is the synthesis phase. We didn't fully look at how glucose is formed, but we got to the G3P, and you can kind of imagine that G3P gets combined with itself to make that glucose. But really, we're seeing how we're able to use that photo energy in order to make these energy intermediates, which are then used to drive the Calvin cycle, which will build glucose, which is what then increases the mass of plants unless we eat them, and then they increase the mass of heterotrophs like you and I. Oh shoot, I need to tell you a password. So this week, the password, I'm just going to grab one real quick, is going to be chloroplast. That's the password for the quizzes and stuff.